today we're working through the whole environmental studies process. We're doing population ecology and why numbers of organisms increase or decrease. We're looking at what an ecosystem is, etc., etc. So this is very basic. It's something you've been doing since you were in grade 3 or 4. It's a lot of it's general knowledge, but you also are going to be learning new terms in line with grade 12. And you must know how to use them, how to apply them, and also all the calculations. There's a lot of calculations in this section of work, and that's how your exam questions are going to be based. They're going to be based on draw a graph, represent this information in a graph, what type of growth phase is this, is it an indirect or a direct method of establishing the population size. And to establish a population size is important because we need to know how to plan for our South Africa and plan for the world. We need to feed people. We need to have sewage and water, etc., etc. And all of this is dependent on how we look after our populations. Also, very often, your questions are going to revolve around animals and the animal world. So think in terms of animals when it's relevant, and in terms of human beings when it's relevant. All right, so population ecology, population size, we're looking at growth forms and the human population. Now, the world consists of pockets of ecosystems within a biosphere. Now, you will recall that the biosphere is everything. It's on what is on land or the earth. Your biosphere is also made up of the air and also the water. Okay, each ecosystem contains various species of organisms, and they are all dependent on each other for survival in some form or manner. Now, should one species become extinct, the others in the ecosystem are affected. And people, this is very important. Because this little line or 2 lines there is what this whole process is about. We need to look after our world. If we take one organism out of the system, it impacts on everything. Now, the survival of all living organisms depends on sustainability. Now, what does sustainability mean? It means to keep the environment so that it is able to support all the organisms that are there. Okay, it must be sustainable. It must be something that can be kept going. That's what the word sustainable means. Of the environment and also the social organizations within a population and a community. Now, social organizations are not just in terms of humans, it's in terms of plants and animals as well. The dynamic balance. Now, remember, a balance is if I've got a seesaw, all right, that is balanced. A dynamic balance means that it will go up and it will go down and vice versa. But at the end of the day, the average is a balance. So we're looking at a dynamic, means to move, balance, to ensure that the ecosystems function successfully and clearly the survival of all species. Now, the community structures within the ecosystem, you must know these definitions. And what I've done is in the X sheet, every single X sheet I've given you definitions that you must learn. Now, it's got lots of reasons for doing that. Number one, if you learn the definition, you know the term, and you can apply it. Also, they will in the exam say, provide the biological term for, and they'll give you a whole bunch of statements, and you must know what term fits with the statement. When you're answering questions and they say, what is ecology? You'll be able to answer that too. So learn those definitions. They will help you. So first we look at what ecology is. Now, ecology, anything with an OGY on the end of the word, tells you that it is a study. So it's a study of the interaction between the living organisms with one another and the non-living factors in an environment. So if we look at ecology, we are studying how living organisms interact with one another and how they interact with the non-living factors. That would be soil, water, air, etc. So the non-living factors, temperature, and the living organisms. So it's an interaction of every living organism on this planet with each other and with the non-living factors. Then we have the ecosystem. Now, an ecosystem implies many things working together. So we're looking at a defined area, a specific area, where plant and animal populations interact. Remember, we're doing ecology, always interaction. So populations interact with each other and with the environment as a community. And then ecological niche. Now, if you find your niche, it means you find your place, the perfect place to be. So generally people that find the niche in for their career, they found their niche. It means that's where their career is. It's their career path from there onwards. They found where they are comfortable. So ecological niche is the status of an organism within a community or an ecosystem, which depends on the organisms. Now look at this. The structural adaptations, in other words, how are they structured? What are the adaptations they have to survive in this niche? The physiological responses, in other words, how do they respond physically? And then also their behavior. So that is the ecological niche. It's the perfect place within a community, depending on the organism's structural adaptations, physiological responses, and behavior. Now remember, and this is just a bit of revision, living organisms in an environment, okay, are called the biotic factors. They are all the living things. And your abiotic factors are your non-living things. That would be the air, water, light, soil, temperature, etc. Remember, these are the things that affect all living organisms. Especially light and water and soil would affect all the plants. If we don't have plants on our planet, people, they are our producers. So if there aren't any plants, well, the herbivores are all going to battle because they've got no food. If the herbivores don't have food, they will die, and the predators will not have, and your carnivores and omnivores won't have any food. So we have to look after every single step within the food pyramid and within the living pyramid of our planet. So remember, biotic is living and abiotic is non-living. Now the interaction, remember, it's an interaction between things, takes place within a community as individuals compete for biotic factors and also food and shelter and space. So these abiotic factors would be your shelter, it would be warm places to live, etc. This will result in specialization within species. Now, if you think about life on this planet, the animals that live in the desert and the plants you're going to find in a desert are very different from the plants that you will find in your garden at home if you live here in South Africa, because unless you're in the Karoo. But everywhere else, we seem to have enough water and we've got very good plant growth and animals will develop nicely. So animals will be specialized to live in areas that are very dry, to live in areas that are rocky, okay, to live in areas that are flat or mountainous. You follow? All organisms will be specialized to survive within an ecological niche. A species, now what is a species? And you have to know this. This is absolutely NB. You must know what a species is for this section of work as well as for the whole diversity section. Okay, species is a group of organisms with similar, now look at this, similar structural and functional characteristics. So similar structure and functional characteristics that in nature will breed only with each other and have a common gene pool. That would be your species. So they have a common breed pool, a gene pool. They will breed with one another and they will look and behave very similarly. Right. Now, basic ecosystem. This again is also revision. We have within our ecosystem the biotic factors, in other words, the living factors, and the non-living factors. Now, these non-living factors will determine which plants and animals and decomposers live in an area, because it would depend on how much air there is, how much water, how much soil, how much, and I remember soil, sure, but we're talking about whether it's rocky, whether it's sandy, whether it's dry, whether it's wet, clay soil, etc. Sunlight, is there a lot of sunlight? You are not going to find the same animals and plant life in a very wet Amazonian valley as you will in a valley that is in the Karoo or in the, I can't think of any specific place, but you follow what I'm saying. A valley is a valley. But if there's a lot of water and there's shelter and there are a lot of plants and animals and they're looking after each other and they're protecting each other, very different from a place where there is nothing. Okay, then we have now, our biotic factors are divided into plants, animals, and decomposers. Our plants are always our producers. They produce, why? They will use the sun, light, and they use water from the soil and carbon dioxide from the air, and they produce food for all the consumers. Now, animals are divided into three sets of consumers. We have number one, the primary consumers. Those would be herbivores. Secondary consumers, generally carnivores, and then we have the tertiary consumers, which are also carnivores in many cases, but they are also omnivores. Okay, population. Now, another definition to learn. Very important. A population is a group of individuals of all ages. Those individuals are all ages that belong, now please, this is important, the same species, that live in the same area and that can interbreed. Now, if you have cats in your house and I have cats at my house, our cats are not the same population. Why? Because if you live in, let's say, KZN and I live in Sandton, very, very long way for those cats to get together to be able to interbreed. The same with, in the Johannesburg Zoo, we have beautiful, beautiful polar bears. Those polar bears are not the same population of polar bears as a group of polar bears that we find in the Antarctic. Why? Because they cannot interbreed. So people, to be the same population, and according to this definition, people that live in America are not actually the same population as us living in South Africa, because it's very difficult to interbreed with someone who lives in America while we're living here. Okay, so think of a population in terms of animals. Always. Try to avoid people because people make it very difficult to understand. So a population is a group of individuals of all ages that belong, and it must tick for all three components. The same species, they must live in the same area, therefore they will be able to interbreed. Then it is a population. Elephants in the Kruger National Park do not belong to the same population as those found in the Addo National Elephant Park. Why? They can't interbreed, people, because they're not living in the same area. Okay. We also get geographical boundaries that will ensure populations remain separate. So let's say there's this huge mountain. The population on this side, so we call them population 1, is not the same as population 2. They may be the same species, but they don't live in the same area and they can't interbreed. Same with a big expanse of water. If you have an ocean and there's an ocean between these organisms, they can't interbreed. All right, they're not going to get onto an aeroplane and fly there. That's why I say think in terms of animals, not in terms of humans. Community. Now, must know a community, also NB. All right, now remember, with a population, it was individuals of the different ages that belong to the same species, that at least belong to the same species, that live in the same area and are able to interbreed. Now, a community is slightly different. It consists of groups of different species that live in the same area. Now, they're not going to interbreed, but they are going to be means between. The word inter always means between. Interdependent. In other words, they're going to depend on each other. Now, an example that I chose here is I said that within a selected community, like the Kruger National Park, the leopard depends on the buck. The buck depends on the grass. The leopard and the buck indirectly depend on the grass. Why? Because they all live in that same area. If there is no grass, the buck will die. If the buck die, the leopards will die. So they are all dependent on the grass in that area. They depend on each other to survive. Without one, the other cannot survive. This is the independence that links all living creatures. Okay, there are 4 main types of interactions exist in a community, and they are competition for food, shelter, space, etc. Predation. Now, predation, let me just straighten this one out once and for all. My cat is a predator, okay, and so is yours. A cat will hunt whatever it can catch for food because it has to survive. But if there are a lot of rats, the cat will then catch prey very easily. If there's only one rat or mouse in my garden, that cat is going to battle to catch that one mouse. So he will not be able to eat regularly unless I feed him. Now, the same happens in nature. You must remember predation, or to be a predator, is an interaction. It is an interaction between, let's say, a lion and buck, or impala. The lion needs those impala because he has to eat and survive. The impala need the grass, so the impala will eat the grass. The grass needs the sun and water and carbon dioxide, otherwise they cannot photosynthesize. So there is an interaction. So 4 main types of interactions. We get competition, predation, parasitism, and mutualism. And we're going to go through all of those next time and the following time after that. Right, population perimeters. What is a population perimeter? These are the factors that are going to affect the increase or the decrease of a population. All right, now, the size of a population is expressed in numbers or biomass. Okay, what is biomass? It's determined by multiplying the number of organisms and the average body weight or the mass of the organism, okay, in a population. So, simple example, there are 10 rats, okay, they each weigh an average weight of approximately 100 grams. That means that the biomass of the rat population will be 1,000 grams, which is 1 kilogram of biomass, biological mass. Population increase. Now, population increase works on birth rate. But birth rate, in life sciences, we use the term natality. Remember we spoke about antenatal classes when a woman's pregnant and she now goes and she goes for classes before she gives birth. Natal means to produce a young. Okay, so natality is the birth rate. And sometimes in an exam or during questions, they're going to ask you about the natality rate, sometimes birth rate. So you must know that the two go together. Okay, birth rate or natality, the natural ability of a population to increase its numbers. Okay, this depends on the ability to reproduce. In the cases of bacteria and viruses, their reproduction depends on whether they're favorable conditions, and they can reproduce over a very short period of time. They can go to huge numbers because they go from 1 becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16, 16 becomes 32, 32 becomes 64, etc., etc., etc. So they can reproduce very quickly because they undergo binary fission, which is a form of mitosis. Right. The higher organisms, we look at a word fecundity, and no, it's not a swear word. Fecundity is the ability of a female to reproduce. So it's the ability to reproduce of any female, to reproduce fertile eggs, which can be fertilized and result in healthy offspring. That's the word fecundity. It's the ability to produce fertile eggs. Okay, now, immigration is also another way to increase the population size. Immigration, look at the I for immigration, is when individuals move from one area into another area, so the I for into. Okay, permanently. It's not temporary, it's permanent. And make this their habitat or make it their home. But immigration, 2 M's, and it starts with an I, and it's when organisms move into an area permanently. All right, so remember, immigration, into. Here is a little bit of a spreadsheet or a schematic so that you can see a population increase, which is positive growth, will be because of a natality or birth rate, which is good, and immigration coming into an area permanently. Population decrease, the death rate or mortality. Now, remember, we talk about us as human beings being mere mortals. We are mortal. What does that mean? It means that we will die. The only thing that you are sure of when you are born is that you will die someday. Okay, everything else, well, it's as fate throws it at you. So, population decrease is due to your death rate or mortality rate. So, death and mortality, the opposite to birth rate and natality. Okay, that's always going to cause a population to decrease. It's expressed in numbers of deaths over a specific period of time. The mortality of a population may be due to the number of factors: lack of food, lack of shelter, lack of space, increase in disease, and predation. Remember, when a population has a lot of organisms and they're all living in one confined area and they start to overpopulate, if one organism gets ill, it passes that disease on to everyone else. It's very quick to then spread the disease. It becomes very contagious and you are able to spread it. So if one person in your home gets flu, guaranteed every member of your family will end up with flu at some other stage over the next 2 weeks. So disease and predation. If your cat has 1,000 rats on your property, it's going to hunt to its heart's content. Okay. The numbers decline because the individuals die and are not replaced at the same rate. Another form of population decrease is emigration. Now think of the E for emigration and to exit. Okay, the E for exit. So emigration occurs when conditions are not good, okay, and the individuals leave the habitat permanently to go to another one. So think of emigration as exit, and immigration is the opposite. Immigration means to go into an area permanently. So the 2 I's go together, and to exit is immigration, and you'll never ever confuse them in an exam. Okay, here we go. Population decrease. We have a mortality rate or death rate decreases, causes a decrease, and immigration to exit will cause a decrease. Okay, migration. Now we've done immigration to come into an area. We've done immigration to exit an area. And now what on earth is migration? Migration is when organisms leave for only a short period of time. Remember, emigration and immigration are permanent, whereas migration is seasonal. All right, so you've got your swallows, your geese. They decide this is getting cold here. And round about when autumn starts, they think, They pack their samis, they pack their bags, hook them onto their backs, and off they fly to a nice warm country where it's the opposite side of the earth, where it is going to now be spring and it's going into summer. So they're actually very clever. They always move at springtime, and at least they move at autumn. They arrive at spring, and then when it gets too cold there, they come back to where they started out. So migration is different from immigration. In immigration, it's the relocation based on temperature and availability of food. And the temperature is going to be based on seasonal changes. your swallows or geese is what I used earlier. They go to a place where it's warm. And here the food is abundant. And when the region starts to cool down, they relocate back. So migration is temporary. Okay, it's temporary. It's only for a short period of time. Okay, population stability. Now, with population stability, when natality is equal to mortality over a period of time, the population is naturally stable. So if we look at your population increase and your population decrease, when they are equal, then the population is naturally stable. It doesn't have to do this on the graph line. It's impossible. What it will do is it will do this. So here there'll be more births, and then there'll be more deaths, and then there'll be more births, and then there'll be more deaths. So what do we end up with? We end up with an average, and the average will be along here, which means the population is stable. It's not going, you follow? It's just making little differences like that. Right. The size of the population can be regarded as stable and constant because of the impact of environmental resistance. Now, the environment is never constant as the impact for food and shelter and disease and predation changes. So, seasonal changes also play a very large role. Animals may die in winter, and that would cause a decrease in the numbers, but in spring the new babies are born, and we then end up with an increase. So people, it all equals out. Okay, that would be a stable population. But if I had a group of rats, I keep going back to these rats. I have something in my head about rats today. But, or rabbits. Let's take bunny rabbits. All right? And there are 50 little bunny rabbits in this field. And 6 females, you know, they have about 8 or 10 babies at a time. So 6 females give birth. And they have, the population goes up by 60. Boom, just like that. Okay? And then maybe 2 rabbits get run over because they try to cross the road. And 10 of them get eaten by hawks. And another 15 of them fall into a big hole and a trap, and they end up dying because their friends can't get them out. Whatever the case is, you're going to find that the population will fluctuate slightly, but that the environment, the area where they live, will determine how many rabbits can live there happily. So if it's a nice big area and there's water and there's shelter and there's space and there's a lot of place to dig their little burrows, everyone's happy. But if that area can accommodate 50 rabbits and there are 1,000 rabbits living there, eventually they'll eat all the food. They'll eat everything and there'll be nothing left over. And they will actually cause the environment to create pressure on them. And that's how we have environmental resistance. It's the amount of food and shelter and water and space that environment can provide for a population. Okay, so environmental resistance becomes apparent as members of the population compete for resources, for food, for water, for shelter, and for space. When the environmental resistance prevents the population from increasing, we say a carrying capacity of the habitat has been reached. Now, to clarify the difference between environmental resistance and carrying capacity. If I have this little hilltop, and let's go back to the bunnies, and this little hilltop's perfect, and it can have very comfortably 10 little rabbits. And these 10 little rabbits hop, hop, hop around, and they're very happy and content, and life is good. Okay? Now, they reproduce. The carrying capacity for that area is 10 rabbits. Not 100, not 1,000, not 50, 10. The minute we have more rabbits, the environment will start to resist for shelter space, etc. Okay? The carrying capacity is what can be comfortably supported in that area. So if you have a 3-bedroomed house, you can comfortably support 6 people. You can't support 50 people in a 3-bedroomed house. Just think of it in those terms. All right, when the carrying capacity The carrying capacity is the number of individuals of a population that can comfortably support it. Carrying capacity changes according to the food supply, competition, seasonal weather patterns, and also the impact of disease. Here you'll see, again, why have these buck come into a place where people are staying? Because there is a lack of food. It's snowing, they're starving, and they're coming into an area that they do not belong in. So this is just the photo to show you that. Okay, the key factors that are responsible for fluctuations of population is rainfall, snow, drought. They are going to affect the population size. If the population density varies greatly from year to year, then it's classified as unstable. In other words, if the graph is doing this. Okay? If the population density does not change much from year to year, it's classified as stable, and you must be able to tell the difference. Okay? Humans or natural disasters sometimes alter that carrying capacity. Fewer organisms are able to survive in a specific area, so we say the carrying capacity has decreased due to the environmental resistance increasing. As the environment pushes this way, your carrying capacity will drop. Fluctuations in populations. The basic food source on our planet is producers. We've gone through this. I'm just going to zip. You've got the food chain, you've got energy transfers, so photosynthesis. We need energy from the sun, carbon dioxide and water, and then clearly mineral salts. Okay? And that limits the food produced by green plants. So in turn, the number of organisms eating those plants will be affected. Density parameters. Okay. There are fluctuations within a population near the equilibrium phase. In other words, it's sitting here. There's your equilibrium phase. And the population will decrease a bit, and it will increase here and decrease and increase. And it all sort of has a balance sort of process like a seesaw. To remain stable, each pair of sexually reproducing individuals must produce 2 surviving offspring. Why? Here we have a male plus a female. Okay? Now, what they need to do is produce 2 offspring, so a little male and a little female, or 2 males or 2 females, so that when they die, they have replaced themselves. Okay. Now, these 2 offsprings, they need to survive for sexual maturity, and that will keep a constant density year after year. Okay. The word density describes the number of individuals in a population in the same area within a specific time period. So we say that's the density then. Factors that regulate population growth, when the population density is high, are called density-dependent factors. In other words, density, always think of the word density as talking about the number of organisms. So when we talk about factors that will affect the population, it is density, so the number of organisms depend on it. It's dependent on how many organisms there are. Density-independent parameters are not related to anything regarding the population size. Your density-dependent parameters would be things like a flood. A river doesn't sit there and think, oh, you know what? There's a whole group of little houses here. Oh, shame. Hey, guys, I'm not going to worry you today. I'll go around you. It doesn't happen, people. So, density-dependent, it depends on the population numbers. In other words, how many organisms are there? And it helps to prevent overpopulation. Okay, so the larger the population numbers, the more the population will be affected. Mortality is increased. Okay, so the population will decrease. The population stabilizes and the carrying capacity is maintained. The need to increase as the population increases and approaches the carrying capacity will then continue. Now, density-dependent factors. People, this is logical. Think again, 1 or 100 or 10 or 100. If there are those number of organisms, will it make a difference? Then you know it's density-dependent. So, food and water, clearly. Competition, space and shelter. All right? If there's no space and shelter, there's lots of stress. Stress causes infertility. Okay, and that will affect the birth rate. Predation. We spoke about the cat and the mice. Okay, if there's more prey, yay. If there's less prey, we have a problem. Okay, disease. Disease spreads quickly when overcrowding occurs. So all of these would play a difference if there was one organism, 10 or 100. Density independent. Now, density independent factors are not related to a population density and are independent of the number. In other words, we are looking at natural disasters. So if there's a veld fire is not going to say, oh, you know what? There are 5 tortoises in this veld. I'm not going to burn here. I'll rather go burn somewhere. It does never do that, people. It's a natural disaster. Okay, is density independent. It happens irrespective of whether there's 1, 10, a million. Doesn't matter. So you've got the flooding of a river. You've got hurricanes. So your density, the density of the population has no impact at all. Biologically, all populations are density dependent because density independent factors will affect the population numbers, causing them to decrease. So population is able to recover after the event, it will stabilize, and it will then grow towards carrying capacity. Here are your factors. Natural catastrophes. Natural disasters is easier to spell than catastrophes. This includes droughts, hurricanes, earthquakes, floods. Population density is reduced non-biologically. In other words, it's not because of a biological process. It is non-biologically. It's the environment. Okay? Climate, when there are severe changes, okay, very cold winter, very hot summers, we end up with the weaker individuals dying. They could either die of a shortage of food, body temperature, etc., etc. I've put in a couple of slides here of a number of disasters so that you can get it in your heads, people, that if it is density dependent, 1, 10, or 100 organisms will have impact. Okay. If I've only made dinner for 2 people and 10 people arrive for dinner, I have a problem. All right. So does it affect it? Density independent, it's natural. It's going to happen. All right. So big earthquake, and that's what it looks like. All these people that were living there, homeless. This is a flood. This flood is not going to say, oh, you know what, look here. There are a whole bunch of people living here and a whole bunch there. Oh, no, man, you know what, we'll just sort of go around. It doesn't do that. Here's another flood. It's happening. All these people can't move. They've got all kinds of stuff. There's a woman sitting on her car, truck stuck in the middle of the road. This is Hurricane Katrina. And you know that they name all hurricanes after women, people. I wonder why. Anyway, Hurricane Katrina. This is in 2005. The wind speeds 280 kilometers an hour. 1,836 people died. Boom. There we go. Direct impact of Hurricane Katrina. This is taken clearly from high up, and it's sort of over the top of this hurricane. The damage, look at this, $90.9 billion US dollars. So we times that by, I think it's what, 7 or 8 now. We've got what it is in rands. Okay. There's a drought. Shame, here's this poor buck and it's dead. It's died eventually of malnutrition. It's got no, starvation, no food. There's nothing to eat there. And no one can control it. All right. Factors that limit population size. All right. Now we've got different stages within their life cycles. Individuals within a population are not necessarily evenly spread throughout the habitat. And this is important. Okay. Evenly spread. In other words, where are they? So throughout the world, there's also a misdistribution or a maldistribution of resources. Okay. And also inefficient use of our natural resources by humans. We mess up incredibly. Now, this can be overcome to an extent when education on correct farming method, pest control, conservation of natural resources, all of this is wonderful and it's all good and fair and well. Okay? If we look here, that's a desert. You can do what you like to that desert. You need to bring water there. Otherwise, nothing's going to grow. Because they say the desert actually has very fertile soil. But it's to get the water there. There is no water. Okay, we look at abiotic factors, people. Abiotic factors are non-living. Okay, so abiotic factors, the rainfall, temperature, light intensity, fertility of the soil, in other words, what is in the soil, mineral content, etc. Animal and human populations are affected by seasonal changes. Now, if we look at biotic factors, your plants and plants are affected when animals and caterpillars eat their leaves. So they're going to have less ability to photosynthesize. Plants are also affected by parasites, fungus infections, diseases, etc. Animals are affected by the availability of food, shelter, predation, parasitism, diseases. What do we want? We have survival of the hardiest and the fittest. And that goes to Darwin's whole theory, that it was survival of the fittest. Okay. Potential lifespan. How long does an organism live for? Okay, so we have, it's not just the number and the natality and mortality rate, but how long they live for. If an organism's going to live for 10 or 15 or 20 years, it's going to have a lot of reproducing to do in that time. So in higher species, mortality is high among the very young and also the very old. Think of us as human beings. Our old people and our babies die. In between, we're okay. This results from predation, availability of food, competition, accidents, natural disasters, etc., especially in the natural environment. Now, the potential lifespan of an organism is usually longer than its actual lifespan. And what do we mean there? The number of years it's supposed to live versus the number of years it actually does live. As humans, we're supposed to live to 70, okay? But some people make it to 100. In fact, there have been a couple. And others die before the age of 30. Now, HIV has made a huge impact on our populations. Population growth and patterns. Now, this is where it is. This entire little section here, very important. Nota bene. Okay, each population shows characteristic growth patterns, which is influenced by the environment. Now, we get the geometric growth pattern. And your geometric growth pattern is an exponential growth pattern. It looks like a J. It's the one where we go from 1 to 2 to 4 to 8 to 16 to 32 to 64 to 128. And so we can carry on and on and on. So in other words, it does that. So I'll do it for you now. So the graph indicates a rapid increase and then comes to an abrupt stop. Okay. Why? Because of environmental resistance. Now, in a bat cave, for example, they'll pollute with feces. There'll be so much feces on the ground, they think, oh, okay, fine, cool, we need to move on. Because they don't exactly have housekeeping. You follow? I mean, they're not going to blips on the ground and then go and get a little pad and put it all into a little bucket and go throw it away. Okay, so they mess up the environment because of pollution of feces. All right, seasonal changes, another one. Antibiotics, medication, people. This is why when you get bronchitis or any kind of bacterial infection, your doctor says to you, take these antibiotics until they are finished. Finish your course of antibiotics. Okay, don't take it for 2 days and because you feel better, don't take it for the next 3 days. Because what happens is this. All of this, the binary fission. Okay, let me just show you here. Case of bacteria, antibiotics, and I'm going to explain to you why. You have a rapid halt. Okay, now, if it's not completed, if just one little organism survives, we go 1 to 2 to 4 to 8, and so the whole thing just carries on again. Okay, so we must make sure that they are all dead. Now, this is a geometric growth pattern. What we're going to have is this. Let me just put here time. Time is always on the x-axis, and this would be your number of organisms. So number of cats, lions, tigers, leopards, bacteria, etc. In this case, it would be bacteria or viruses. So your geometric growth phase would start with one little organism, okay? And in fact, let's make that 10. So it starts here at 1. and it will just go slow increase, and then all of a sudden it just goes sugar and it goes up. Okay? At this point, they have now reached absolute environmental resistance. They cannot survive. That's it. Overs, cadovers, and they all just die. Or you've taken an antibiotic. Now, if just one, only one needs to survive, and watch here, we start the whole process again. And how many times have you heard people say to you, this is the third set of bronchitis I've had this winter? Yeah, it is, because you are clearly not taking all your antibiotics, or you're not taking them at the exact time every day, or you are not actually looking after yourself. Okay, 99% you haven't taken the antibiotics when you should. All right, logistic growth pattern. Now this growth pattern can also be called the sigmoid graph. Okay, so we have geometric growth rate with a J shape. Here we have a sigmoid growth with an S curve. Now, this is how it will look. We've got 5 phases, so let's just zap this up again. So we're going to put all the time along here, whatever the time is. Remember, I'm just doing a basic graph pattern. And over here we have our number of organisms. Okay, what happens here is we start off, we're going to go to our bunnies. And as I talk about the bunnies, and I'm going to tell you a beautiful little bunny story. It's a pity it's not Easter, okay, but a pretty little bunny story. We have 10 little bunnies. And these 10 little bunnies decide one day they want to leave the farm, and they go for a walk. And they walk and they walk and they walk and they walk, and they find this beautiful place to stay where everything just works beautifully. And they are living nicely and happily, and they mate off, and they dig little burrows, and the females get pregnant and they start giving birth. And as they give birth, 2 little bunny rabbits get caught by a hawk, and another one falls into the water. And so and all these things start happening to our little bunnies. So over the period of time, we have an S-shaped curve. There's my S-shaped curve. Okay? During this S-shaped curve, we are going to have phases. So this phase here is when they are acclimatizing. This is the lag phase. So at this point, our little bunny rabbits are acclimatizing, they're mating, the females are pregnant. Then we have geometric growth phase. This is the same as the J-shaped graph. So this is the geometric growth phase. What happened here? Those 5 females, because remember we started off with 10 little bunnies. We started off with 5 males and 5 females. Okay? They mated. Here, zhoops, we've now gone from 5 rabbits to 60 rabbits or 70 rabbits. Okay, that's the geometric growth phase. Then it starts to slow down over here. We're starting to reach an equilibrium phase. Okay, so these are your phases. Now here we have a stable population, but let's say something happens and a disease hits these poor little bunny rabbits and they go to here. This would then be the death phase. And these 2 phases in between here, there, I'm going to go show you now. So we have the initial lag phase. Okay, this is when they acclimatize, they are busy reproducing, there's gestation takes place, and everything is wonderful. They dig their little holes, little homes, happiness is. We have the accelerated or the geometric growth phase. This is now maximum growth rate. Okay, little or no environmental resistance, natality rate is high. 5 females all give birth to 10 babies. That population is going to go from 10 plus 50 babies puts them to a population of 60 in one mating session. Okay. Then we have the decelerating growth phase or deceleration growth phase. In other words, it's not like the geometric growth phase. It comes up, geometric growth phase, and then it starts to slow down. As it slows down here, it has decelerated the growth. So it slows down. Why? They start competing for food and shelter. And there's increased predation. Those hawks sit there and they check out and they think, ha 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 ha, 60 little rabbits running around. Let's see who can get out of my sights here. And they go, zip, and they grab a little rabbit. Okay, that's predation. Little fox comes along and says, check this out, guys. Look at all these little rabbits. They think they've got carte blanche. They don't know that we are the predators. We are in charge. So little fox takes out his cell phone and he dials the next little fox and says, hey, dude, you know what? There are like 15 to 20 rabbits here on this hill. Come, we've got mealtime. We're going out to a restaurant tonight. And that's how it happens. So the other fox will immigrate into that area so that they can now live there because there's lots of food. So predation increases. Parasitism. If one of those little animals gets ill, they spread it because they're all living close to each other. So the birth rate or natality rate is still higher than the mortality rate. And then we reach our equilibrium phase. Everything is fine. It's stable. It will vary slightly, but it's stable. Why? We have a carrying capacity of the environment, and that carrying capacity is about there. We've reached the carrying capacity. And then the last phase is the death phase. So in some cases, the growth indicates a dramatic drop, and that's when we have death and causing of extinction. Why? Extreme environmental resistance. Remember the picture I showed you earlier about that buck lying in the middle of nowhere with a drought taking place? That's the problem there. It's drought. That's environmental resistance. The environment's saying, I cannot look after you. I cannot provide food, water, shelter, and space for you. And the animals have a choice. They stay there and die, or they move. Okay, question 1. The table below shows the population of rats in a millie field over a period of 10 years. So you're going to have to draw a graph, and that will be the title of your graph. So it's the population of rats in a millie field over a period of 10 years. There is your heading that you are going to put into this graph. All right, now what we've got here is, I've done the graph basis here already, but we've got the population and we have the year. So we started 1997 and we go to 96 on the x-axis. This is time. And put it in because it is a mark. Okay? On your y-axis here, this is going to be the rat population numbers, or rat population, number of rats. Something in that line is going to be what you write on your y-axis. Okay, and we've got our heading, which is again another mark. So I haven't written it out here, but you know what the heading is, so there's my heading. Now I've got to use the table above to draw this graph. So we're starting at 30, 40, 80, 130. So let's go each of the years. So that would be 87. This would be 88, would be about here. 89, 90, 91, 92, 93, 94, 95, and then we go to 96. All right, so we had the first one was 30 in 1987. What was 30, 40, 80, I think? 30, 40, 80, 130. So 30, and the next year is 40, and in 89 was 80, 130, 140. In 1990 was 130, 130, and 91 and 92, so we go back to 130, so we go 140, back to 130, 95, 115, 95, and then it starts to go up again. So that would be 110, so 115, and then it drops back to 110 and 110. So this is our graph. Okay. Okay, people, so there's your graph. So now let's look at our marks. Okay, they're going to say, is this the correct graph? So did you draw a line graph? Yes, you did. So it's the correct graph is your mark. Did we put a title? Well, I didn't write the title here, but there's the title, the population. Your title's always in the question. So the population of rats in a mealie field over a period of 10 years. So we've got our title done. Okay. Have you labeled the x-axis correctly? Yes. Have you labeled the y-axis correctly? Yes. Have you got the correct units here? Is it the correct spacing that you've got here? Yes, it is, because it's made my graph not be a lopsided graph. So I've gone 0 and then 10, 20, 30, 40, 50, and up to 140. So have I got the correct? Yes, I have. My years? Yes, I have. Okay. Are the different points, have I got all the points correct here? So what they'll do is they'll say each of these will be a mark. So did you do your intersects right? Yes, I did. So it's 1, 2, 3, 4, 5. So it's 1, 2, 3, 4, 5, 6, 7, 8, 9. And what they'll normally say is if you've got 1 to 2 points correct, then you get a mark. And so we carry on. This would be a total, I've got them all correct. It would be a total of 5 marks there for having all these points correct. Okay, so if we add all of this up, then you've got the heading. 2, 3 plus 5 is 8, 9, 10, 11, 12. A neat 12 marks there for your graph. Okay, people, what type of growth form is shown by this graph? Now look at our graph. Okay, look at our graph. We've got slight lag phase, geometric growth phase. Okay, it reaches carrying capacity, so we have a deceleration phase. Then we have it reaches capacity, it decreases and increases, and here I have my stable population. So it's rats. Remember that when we look at bacteria and viruses, we're looking at geometric. So this would be a logistic growth pattern or a growth form. You can call it a pattern or a form. Okay, if we look at C, indicate the carrying capacity of the mealie field on the graph. Now, if we look at the carrying capacity, the maximum carrying capacity of this mealie field, all you have to do is, you don't even look at the graph, look at the table. It goes 130, it goes to 140, and it comes down to 130 again. And it doesn't go back to 130. So we've reached the carrying capacity there. All right. So your carrying capacity on the graph, as far as this experiment is concerned, is a carrying capacity maximum of 130 organisms. So 130 what? 130 rats. Why? We're talking about rats, people. So don't just write 130. Don't ever forget to put in the unit of whatever it is that you're working with. List the abiotic factors that affect the carrying capacity of an area. This you shouldn't even have to think about or learn. It should just be there. It's your soil. water, okay, temperature, light, humidity. Because remember, humidity is also going to affect things. So soil, water, temperature, light, humidity, and it's not just light, it's the intensity of the light as well. So there you go. There are 5. They wanted you to list 5. That's it. All right, determining population size. We're going to have to go quickly now. You've got, it's based on qualitative and quantitative. So what is the quality of the samples and how many are there? So it's qualitative and quantitative data that we're going to get on all the animals and plants and things in the abiotic, as well as the abiotic environment. Now, based on these findings, we monitor, evaluate, and manage the environment and the populations that inhabit them. So, if, for example, you and I had a game farm, okay? On this game farm, we need to know how many impala do we have? How many springbuck do we have? How many gazelles do we have? How many lions do we have, if we're lucky enough to have lions or cheetahs? How many crocodiles do we have? We need to know this. Why? So we can look after that environment. We don't want the elephants to outbreed everything and stomp on everything, but we also don't want our buck to eat everything. If there are a whole bunch of impala and they eat all the food, there won't be anything for the gazelles and the springbuck. So we need to manage. We need to monitor, evaluate, and manage the environment. Okay, results on the findings are used to do survival strategies and also to manage those changes in the population over time. Okay, first we have to determine the numbers of the population. We have to look at the distribution of the individuals. Now, there are 3 different types of distribution of individuals in a population. We have regular distribution, and the regular distribution shows the normal even distribution. So this is what we have here. And you're going to have your animals marking out this little territory saying, listen, this is my area here. The lions do it, the leopards do it, the cheetahs do it, birds do it. Birds sit on a wire and they sing and say, this is my area. But we're looking at regular distribution. So regular distribution would be something like this. Okay? They distribute it evenly throughout the area. Regular distribution, not difficult. Then we have random distribution. So here there's no territoriality. It's just organisms live where they live. Okay, so random would be random, anywhere. And our third type of distribution is a cluster distribution. So you would have a group here, and it's pretty much like us human beings live. Another little group here, there's another cluster of townhouses there, another little cluster of townhouses there, maybe one of here. So that would be cluster distribution. And this is a trend for social animals. Okay, your pride of lions, elephants, etc. They will sit and they will go to an area where the group can survive. All right, so that's how they'll be distributed. All right, an investigator does not use the same method to determine the numbers of weevils in a bag of flour, which is horrible, okay, as the elephants in a Kruger National Park. You estimate those weevils because you're going to have thousands and thousands, whereas you can count your elephants. So we look at and people, this is important, the choice of method that you use, and this is what they will give you in an exam, is the accuracy that's required, the type of organism you're dealing with, and the habitat that's involved. If you want to count the number of tigers that are, really think about it. If you want to count the number of tigers that are in the Amazon, and you can't see through those trees, and you have to like chop half of your way through the pathway through the jungle, it's going to be very, very, very difficult. You're going to use a very different way of counting the animals there or establishing how many there are as you would on a sheep farm in the Karoo. So you've got to look at accuracy. How much accuracy is required? Well, in any research, we would hope the accuracy is very good. Type of organism, is it a little weevil or is it an elephant? Okay. And the habitat. Can we move through that habitat? Now, methods to determine population size. We've got the direct method. Now, this is easy. When an organism is large, we can count them. We can say, hang on. I saw 5 elephants today in that, I can't even think what they're called, the group of elephants. Okay? And they don't move around too much. But have you ever tried to count sheep? That's why they tell you to do that when you can't sleep. Okay, because they move around all the time. So each individual of the population is counted, and we call this the census. Now, if you remember a couple of years ago, we had, I think in about 7 or 8 years ago, we had the census in South Africa. And you had these chaps and women and men coming around to your home, and they had like a document that you had to fill in. And on that day, at that time, how many people lived in your home? And they then go back, and based on that, the government will then check out the needs of the people. And they say, hold on, we need to have more homes, better health care. There is only a clinic 20 k's away, so let's put a clinic closer. What water and sewage do we need? Everyone that lives in this little area here has children that are in little. They're like under 3 years old. So maybe we should look at putting primary schools and nursery schools in place. And they use it to look after the community. Now, when counting animals, we define an area and we count as quickly as possible. If the area is too big, or the animals mill around and move, we can use aerial photos. So there, you'll take they're specially designed aeroplanes, and they go up with a camera, and they take photos of an area of about a kilometer and a half wide, sometimes even further apart, depending on how big the animal is you want to count. Kilometer wide for elephants, but if you're going to be counting impalas, maybe only 500 meters wide, and then you print out the photos and you can sit and you can actually count the individuals at the same time. All right, so aerial photos. Game rangers can tell the number of populations by counting footprints and also animal droppings. I've never understood how they can do it, but they can do it. They are very accurate. And then we use the census for what? When the animals are large, like us human beings, when they inhabit a fixed area, like we do, and the elephants in the Kruger Park, and they can be counted quickly and easily. That's the big secret there. Now, when you can't count something because it's not big enough, it's smaller, or the environment isn't right, etc., we now go to the indirect technique. Here we're going to estimate, not guesstimate, estimate the population size. Okay? The individuals must be counted accurately for each sample, people. All right? And then we take the total and we work it out with a very, very simple formula. Now, there are 2 types of direct methods. There's the quadrant method and there's the mark recapture mark method. Now, quadrant means square. It's a quad, is a square. So, the quadrant method is actually very easy. This is if we have a large number of organisms or a very big area. It's used to determine the species density, in other words, how many different species are living in an area, okay, at one time. So the species density, means the numbers, is found in a given area, and we compare those results to other areas and other species. So let's say we want to find out how many impala there are in the Kruger National Park versus the Addo Elephant Park. Because not just elephants, they could be there as well. Okay? We look at the species density, the numbers. Density always means numbers, remember that. Okay? The species frequency, in other words, how many are there? Are they in groups? Are they randomly spread out, etc.? And we look at the quadrants. So now, the quadrant method is very easy. You're going to take 4 nails or 4 sticks, and you're going to measure out a 1 meter by 1 meter square area. You put a piece of string around it and you count whatever's in there. So 10 blades of grass, 15 weevils, 10 little beetles, 5 lizards, 2 snakes. and you write it all down. So the problem here is that you have to be very careful. You've got to select the quadrant at random. You use nails and string to mark it out. You repeat it for approximately 10% of the total area, and the accuracy is very important for each one. So if I'm looking at earthworms, I must make sure I count every single little earthworm that could possibly be in that square meter. All right, now you use the following formula, and this is easy and you must know this. This is NB. They're going to ask this to you in an exam. All right, I'm not sure why those letters are smaller than this, but we're going to improvise here. We get the established population, the numbers in the sample times the total size of the area, and that will give you the size, and then divided by the size of the quadrant. All right, now the size of the quadrant will be a meter. So let's have a look. This is how the actual calculation goes. Okay, we're going to be looking at samples. So we're going to be looking at animals, organisms, let's say locusts. So in this quadrant, we had 1. In this one, we had 2. In this one, we had 6. In this one, quadrant 8, and 4, and 3, and 2, and 1, and 2, and 4. Now we add them all up, it gets to 33. There are 10 quadrants here, a total, which is, I divide by 10, and that gives me 3.3 rounded to the nearest number. So let's say I was looking at locusts. I can't cut the locust in half or in a third, so I round it off. I'm going to end up with 3 locusts on average per quadrant. Okay. There's an average of 6 crickets per 0.1 meter squared, so that's 0.01 meters squared, with a total of 1,000 square meters. The estimated population would be 6 crickets times 1,000, the total area, divided by the quadrant size is going to give you 600,000 crickets. Okay, there's 6 crickets per 0.01 meter squared. Okay, 6 times 100 to make 0.01 equals 1 meter squared. So we end up with approximately 600 crickets per square meter. Because remember, there are 600,000 in that total area. We want to figure out how many there was in one area or 1 meter squared. Therefore, in 100, because you've got to know the overall number, 1,000 meters squared, we're going to have 600,000 crickets. The reliability, people, very important. Quadrants must be selected at random. Okay? Each individual inside that quadrant must be counted accurately. There's no skimping here. You have to count whatever the sample is very well. Quadrants must cover at least 10% of the total area. So if I have a total area of 50 square meters, I must make sure that I cover 10% of that, which means I'm going to have to cover 5 square meters. All right, always 10%, and you must know the total area. So 4 conditions: random, accurate counting, you must cover 10% of the total area, and you must know the size of the total area. Okay, now we go to the mark, recapture, mark method. Now look at the name of it, people. It says it's the mark, recapture, mark method. So we have a farm, we have our game farm. We want to know how many impala we have on the farm. We say, okay, we can't go around our farm and count every single impala. It is impossible. There are too many. So we catch a whole bunch of impala. We mark them and we let them go. After a period, 2-3 weeks, because we let those that we've marked go so they can mix with the others. And then we collect them again. We catch them and we bring them into the enclosure. When we get into the enclosure, they're going to be animals that are marked and those that aren't marked. The ones that are marked, we caught in the first one. The ones that aren't marked are new. And then there's a formula. Now, this mark, recapture, mark method is also called the Peterson's Index. Okay, and it's used to establish estimated population size of animals that don't remain in the same area and are not always visible. Now, they're not always visible because they're camouflaged and they move around. So if I have this huge Kruger National Park, okay, you're going to have some animals here and some there, and then those there and those there. So these are going to move to here, and those will move to here, and some of them will move there, and others will move there. So it's when they move around, you can't keep track of them. The area is too big. Using this method, we catch, mark, and release the animals, okay, and then we catch them again. So after they've been allowed to mix with the rest of the population. Remember, population, same species belonging to the same, animals of different ages belong to the same species that live in the same area that interbreed. Okay, so there'll be marked and unmarked animals in that second catch, and we use the following formula. You've got to know this formula, all right? N equals C times M over R. I'm not too concerned about the letters of the alphabet. What I am concerned about is that you know it's estimated population. We take the total number in the second catch times the total number in the first catch. So if we caught 50 in the second catch and we caught 30 or 50 as well in our first catch, and then we look at the number that are marked here, and the number that are marked, let's say there are 20 that are marked, you can superimpose these two numbers as well. It doesn't matter. Whether you put total of first catch versus total of second catch, this times that, it doesn't matter which way you put them around. The reliability of this method improves each time you recapture and release these organisms. So we take the total of the third catch times the total of the second catch, divided by the ones that were marked in the second catch. Because remember, the third catch, because these that are marked here are the ones that were marked in the first and the second catch. Okay, the more times the steps are repeated, the more accurate it becomes. So here's a question, and we've got very little time, so we're going to work through it quite quickly. We have a game farm, people, and we want to establish the number of impala. You and I. Okay, we got a partnership. It's like this TV program we have. Okay, on a set date, we say, fine, we build our enclosure, and we go and we set up on horseback, and we round up our impala. We managed to round up a whole 50. Once they're safely in this enclosure, we mark them and we set them free. After 2 weeks, we set out again, so we give them 2 weeks to mix. So we caught them, we marked them, we release them, now we go and we catch them again. Catch our second lot, and we only catch 42. Among the animals in the second catch are 10 marked animals. So we caught 50, we caught 42, our second catch, and there were 10 that were marked. Okay? Calculate the estimated population of impala that the farmer has on his game farm. Show all your calculations. So, okay, here we go. We've got N equals C times M, or it can be M times C, doesn't matter. Okay, I'm putting this here not as a mathematical thing, but you can move it the other way around, divided by R. So we're going to look at, in our second catch, if we go according to this, in our second catch we had 42 animals, times the ones we had in our first catch, which was 50, divided by 10 that were marked, okay, is going to be equal to, where's our calculator? Okay, we've got 42 times 50 is equal to, okay, which is, oh man, which is, and I've two, I'm not going to write it out here. So we got 2100, and we're going to divide that by our 10 is equal to 210. So the answer here is 210 what? 10 impala, because that's what we wanted to know. Now if we release them, we're going to take our, this was our second catch times our first catch, then we'll take our third catch times our second catch. And the ones that are marked will be the marked in the first and the second catch. So it will all work out very nicely. Okay, people, remember, you must always mark the individuals in a way that's not going to harm them. In other words, you're not going to take a lion and paint a huge, big, yellow dartboard kind thing on his rump. Okay, you're certainly not going to take an impala and put a beautiful, bright red, broad band studded collar on it. You're not going to take a hawk or an eagle and put a beautiful bangle type ring on it. So you're going to mark those animals in a way that is not going to disturb their normal way of living in any form or manner. No bright colors. If you're going to, maybe just put a clip into the ear, but nothing that's outlandish, and you can't use paint that's got lead in it, or that's going to wash off. So make sure that whatever method you use to mark the animal, it's logical, it's going to last for the amount of time that you release it before you recapture it. So you can't use water paint and it rains and then everything washes off. So use your common sense there, but you must be able to think about this in an exam because they can ask it. Okay. It's also important that the method of marking stays visible, so it mustn't harm them. It must stay visible. Okay. And if we look at the different methods of tagging, paint must be non-toxic and it must be waterproof. All right. You use that in snails and tortoises. Then rubber rings on the legs of birds. And those rubber rings mustn't fit too tightly. So if this is the poor bird's foot, and you take this rubber ring and you put it around here and you cut off its circulation and its foot falls off. Okay, but it mustn't be too loose, because if it's too loose, it's going to hook on things and you're going to end up hurting the bird. So it's got to fit snugly. Plastic bags, plastic tags, through the ears of a buck. So if this is the little ear like that, and you go and you get a little tag, and you just put a little tag on like that, so it gets a pretty little earring on the one side. Branding an emblem on the animal's rump. They use that on the farms, in the cattle farms all over the world. Okay, get this red hot iron, you heat it on a fire, and you just jab it and you singe it, and you actually scar the poor animal. So it's sore for 5 or 10 minutes, I'm sure. But you put a branding on it. Shaving hair in a distinct pattern. I don't know if lions and those kind of animals will like that very much. They get a bit of a hair trim, but also it can be a way of doing it. Aluminium discs on the operculum of fish, sort of like putting a nose ring or an earring in. Now, we can only use the mark recapture mark method. Now, please, this is important. Population numbers do not change rapidly. Okay, you can't do it on fleas and flies, on aphids and little things like that that are reproducing quickly. The numbers can't change rapidly. Okay, they've got to at least stay constant for a couple of months. The area is closed. In other words, the area must be closed. You can't have immigration and emigration happening. It's got to be like the Kruger National Park. It's completely enclosed in. You can't have you got a fence around here, like a ranch, like a game farm. So no organisms can immigrate, and no organisms are going to emigrate, to exit. Immigrate, to enter. The individuals have a lifespan which is long enough for you to capture, mark, and release them. You can't do it on a fly, people, because if you have a fly, what's going to happen? You're going to catch it, you're going to mark it, you're going to release it, it's going to be dead before you catch it again. So you've got to make sure that the lifespan is long enough for you to actually mark, recapture, mark. Okay. The recapture process must be completed in a period which is long enough to ensure that the marked animals have a time to mix. Because it's no good you only catching, if we go back to the circle here, you're only catching the animals that are in this sector here. You've got to let them go, and they've got to be released so that they can go mix with these, and these guys can come here, so that when you catch this lot here, you've got a mix of everything that was in the enclosed area. Okay, so they must have time to mix with the rest of the population, and it must be not too long, because if it's too long, This is like Goldilocks and the Three Bears. If it is too long, what are you going to end up with? You're going to end up with a whole bunch of births having taken place and a whole bunch of them having died along the way from old age before you actually got your act together. So you need to look at the animal and what method you are going to use to establish that specific animal. All right. Now, let's just recap where we've been today. We're looking at population ecology. There are terms that you have to know, terms and definitions. Ecology, ecosystem, ecological niche, a community. The difference in a community and a population. Population, same species, same area, interbreed. A community, different species that live in the same area, but they're not going to interbreed, they're going to interact with each other. They need each other to survive. Then we look at the different ways of different approaches to a population. Density dependent depends on the number of individuals. Do I have one person over to dinner, 10 people, or 100 people? My catering is going to be very different. Okay, for food, shelter, space, and mates. Okay, then density independent. Okay, it's independent. Is going to be things like natural disasters, veld fires, earthquakes, volcanic eruptions, floods. They happen. Doesn't matter how many organisms there are. It's density independent, people, independent. All right, and I'm now running very low on time here. Then we've got direct method, census. You can count. Indirect method, we've got quadrant. You mark off a quarter, 1 by 1 meter. It must be selected at random. It must be very accurate. It must cover 10% of the total area, and you must know the total area. Those are your 4 conditions. Then mark, recapture, mark method. You go, you catch the animals, you mark them, you release them, get them to mix, and you get them back. Okay, and then there's a calculation. Estimated population is the second catch times the first catch over the number of animals that are marked in the second catch. Why? Because they come from the first catch. Okay, and that is it for today. We got through it. I spoke a lot. I spoke quickly, and I tried to get everything onto the screen that you needed because I knew I had to work through this section very quickly. Next time, we're going to carry on with the whole social groups and social interactions of organisms, etc., etc. Until then, have an awesome, awesome time. Study hard. The effort you put in now, you will reap at the end of it. So there you go. Have a good one.